Hello, everyone, and welcome to a special CERM event marking Stem Cell Awareness Day. We've got a jam-packed agenda today, so we're going to jump into it right away. We're going to be talking, first of all, about COVID and the steps that CERM has been taking over the last few months to try and respond to the crisis, to the pandemic. And then a little later, we're going to be talking about some restrictions on research that could impede the ability of the whole field to, sorry, that's me talking to myself on YouTube. I'll stop that. Sorry, technical difficulties. This is a whole new adventure for all of us. So there's going to be some glitches, and I hope you'll bear with us during that. And in the meantime, I'd like to introduce our first guest, which is the chairman of the CERM board, Jonathan Thomas. JT, can you hear me?
I can.
Great. Well, welcome to this special Stem Cell Awareness Day event. And let's start off by talking about COVID-19 funding. A few months ago, the board decided that they had to kind of jump into this and put up $5 million in emergency funding for COVID research. What was the reasoning behind that?
That's right, Kevin, and welcome everybody. It's a pleasure to be able to speak to all of you today and to give you a bit of insight into our process and thinking. So when the pandemic hit in earnest in early to mid-March, we started having internal discussions about the possibility of being able to fund some research connected to COVID. And as we were thinking about that, we began to get wind of a number of our scientists who indeed were working on that sort of thing and felt that that would be a real way to jumpstart any sort of program to fund COVID research. We felt absolutely that because literally the entire world was committed at that point to collaborating on finding treatments and cures for this condition, that it behooved us to act. CERM, as you know, for many, many years now has had an infrastructure in place that allows it to go out and solicit bids for research and to evaluate them through expert peer review and to put that sort of thing into place in a very quick order. So middle of March, we had some internal discussions. It was on a Saturday, remember vividly, and I spoke with our CEO, Maria Milan, about this, and we decided we wanted to bring it to the board to have it as the subject of a potential $5 million round, which at this point was all we could afford to put into it. So in that discussion, we decided to move forward, and the team was activated. And I'm very proud to say that within 6 days, we had a board meeting to discuss the concept of funding and an RFA, or request for application, to go out to members of the science community in California for projects related to that disease. Over the course of the next 10 or so weeks, we literally had 10 separate board meetings on this topic. We had 7 separate peer review sessions on applications that were submitted, and things moved like clockwork in a very expeditious fashion, which ended up with us funding 17 programs, 3 of which were clinical trials. So we were very proud of the fact we were able to contribute. And as we were seeing now and getting progress reports on a number of these pieces of research, things are moving along, and there are discoveries being made every day in this, further to what we've enabled.
It really did move very fast, as you said. I mean, literally within a few weeks, we had an application that we were funding for a clinical trial. I will be hearing more about that in a few moments. Were you surprised how quickly it worked?
Not at all, Kevin. I think I can safely say CERM is long since a well-oiled machine in getting best-in-class grants out to deserving scientists throughout the state. So once we decided to put this out there, we had no doubt that we would get the highest quality applications coming in, and our peer reviewers, who are key to the whole process, which for those of you who aren't aware are picked from a pool of stem cell experts literally from around the world, all of them being outside of California to avoid any particular conflict. They were, as they always have been throughout CERM's existence, very eager to contribute. So they jumped in and were meeting almost weekly on these applications to make this happen. There was no question that there was an incredible sense of urgency and unmet need here. So everybody pitched in and all virtually, as you might imagine. This was all done over Zoom. And it really went like clockwork, and we were able to meet the challenge and get these grants funded in a very quick period of time, much quicker, I would add, I believe, than most any other grant making entity could have pulled this off.
And with this round of funding, the board also instituted at the behest of one of our new board members, Isabel Duran, new requirements as well. So could you talk a little bit about those?
Certainly. So one of the things we wanted to make sure was, given in particular that COVID was disproportionately affecting underserved communities, we wanted to be certain that all of the research that we had took that into account. And in fact, as part of their written submission, had a plan that would demonstrate that they would be able to factor in incorporating underserved communities into the studies in the form, for example, in clinical trials, of making sure that there was a wide demographic representation in the patients who are going to be subject to the clinical trials, that sort of thing. Our board member, Isabel, has been a tireless champion of this notion, not just for COVID, but for every disease that we enable through our funding, and has been a great spokesperson and is driving us to go even further than we always had in making sure that diversity is taken into account and underserved communities are served.
Great. Thank you, JT. We're going to move on now to talking about some of the clinical trials we're funding. And I would like to bring into the discussion now Dr. Sohail Talib. We're delighted to have you here, Sohail. How are you doing?
I'm doing good.
Good. Dr. Talib is one of our senior science officers. He works on a lot of the clinical trial projects that we fund. He's also heavily involved in our business development group. And before we begin, I'd like to show a video of one of the researchers, John Zaya, who was the first one who we funded for a clinical trial.
This project focuses on an evaluation of SARS-CoV-2 antibody in convalescent volunteer donors. And the goal is to assist medical centers where COVID-19 infection is most active and help them obtain convalescent plasma. In addition, assess what effect the plasma has on recipients in terms of outcome. We're using antibody measurements of various kinds, both binding antibodies and functional antibodies, namely those that can neutralize the virus. And we hope to assess whether or not these levels of antibody have any relationship to the outcome in the patients. Thank you.
So Sohail, we've heard a lot about convalescent plasma. It's been in the news quite a bit lately, not surprisingly, because of its potential to help people battling the virus. But Dr. Zaya's work is slightly different in some respects than some of the other projects out there. Could you explain how that is?
Yes, Kevin. So I think this study is trying to overcome some of the challenges with this type of treatment, which includes logistic challenges, that is collecting plasma from the patients, specifically the patients from underserved communities, as well as related to the quality of the plasma that's being used for the treatment. So specifically, Dr. Zaya and his team is working to identify the key active ingredients in the plasma that's being given to the patient. So the blood samples from the patients who receive these treatments will be tested before and after the treatment. And by doing such analysis, Dr. Zaya and his team will be able to find out which active ingredient from the donor plasma is responsible for the treatment effect. So basically connecting the dots. What was given, what was the active ingredient was, and what made the difference. So by doing that, the future treatments will be more precise and will have a greater chance of success.
So in a sense, by doing this kind of detailed analysis, we get a better sense of what are the active ingredients in the plasma that can help people?
And that's exactly right. Yeah, that is the idea.
Okay, great. Okay, let's move on to our second clinical trial, and this is with Dr. Michael Mattei from UC San Francisco.
This project focuses on an event.
My apologies. I told you there'd be glitches, and I don't disappoint. Let me see which one.
Hello, I am Dr. Michael Mattei. I work at UCSF, and I am very pleased to thank the CIRM organization for awarding us a grant to allow us to carry out a clinical trial to treat patients with acute respiratory failure, primarily from COVID-19 infection. This grant will be very important to allow us to expand to the trial to the University of California at Davis, as well as continue our trial at UCSF San Francisco and Zuckerberg San Francisco General Hospital. The treatment that we are testing is mesenchymal stromal cells, a cell-based therapy that has considerable promise based on preclinical studies, and we also know it has an excellent safety profile based on clinical trials we've completed. So we are in the early phase of carrying out this 120 patient clinical trial. And again, very much appreciate, I do and my colleagues at UCSF, the funding of this grant by CIRM, hoping that this will improve clinical outcomes for patients with acute respiratory failure in the intensive care unit from COVID-19. Thank you.
So Sohail, it's an interesting work. I mean, can you talk a bit about the cells that he's using and why we think that they may be beneficial in this kind of approach?
Yes, exactly right. So the type of the cells which Dr. Mattei is using are called MSCs, mesenchymal stem cell. And this type of the cells, Dr. Mattei, as he described, has a long experience of using these cell therapy for the treatment. And he has decades of experience in using these cell therapy in the patients which had acute lung injury. And these types of cells are known to have a calming effect on the immune system because some of these patients have an overactive immune system, which is ravaging the lungs of these patients. So by giving these cells, which can calm down the immune system, overactive immune system, has chance of potentially repairing the damage which has been done to the lungs of these patients. And I think Dr. Mattei is very well known and well recognized in this field. So he's building on his own experience from the decades of work which he has already done using these type of cells.
Great, thank you. And he's already using this approach. He was using this approach in other clinical trials beforehand. So what added value does our support for him, our funding for him, bring to this?
That's exactly right. So Kevin, I think this is something which you pointed out, is that what the added value of this, because it's a multi-center clinical trial, but what it really importantly, this grant allows Dr. Mattei to extend this clinical trial to the areas of California in which therapy will be available to the patients from the underserved community. For example, he's extended his clinical trial to UC Davis. So all the areas around UC Davis, Sacramento, and on that area, which has a large population from the underserved community, will be able to benefit from this clinical trial.
Great. And the third clinical trial we're funding is with a company called Cellularity. Can you talk a bit about that, please?
Oh, sure. So Kevin, this is the goal of this particular clinical trial, which is carried out under the leadership of Dr. Zhang, is to boost the immune system of the patient. So the healthy individuals, the type of the cells which is called the natural killer cells, are the first line of defense against the viruses. So they rapidly detect and kill the virus, and thus they stop the spread of the virus to other cells. So that is the normal mechanism of the immune system, which is present in the healthy individuals. But what is known that the patients which are infected with the virus, that's the coronavirus, and they have a reduced number of these cells, the so-called natural killer cells. So the body is unable to fight the infection. So the hope of this clinical trial is the administration of these NK cells will boost the immune system and of the patient, and thus will be able to get rid of the infection.
Great. So these trials all have a number of things in common. Obviously, one is our funding. Another is it shows our ability to bring these trials to California. But the third is also the fact that they're all being held at CERM alpha stem cell clinics, part of a network of clinics around the state that we help create. Why is that important? Why is what advantages does it have to put those in those clinics?
Oh yes, I think that's a very important point. So the alpha stem cell clinics were set up by CERM to accelerate the development and delivery of the regenerative medicine treatment to the patients. So all these three clinical trials which we discussed today are being carried out in these alpha stem cell clinics. And they provide a unique advantage by connecting connecting these investigators who are doing these clinical trials to the medical centers which are expert in these areas, which includes UCLA, UCI, the University of California, San Diego, City of Hope, UCSF, and UC Davis. And more importantly, the outreach to the patients from the underserved communities, so that these treatments can be carried out very effectively and can proceed without any delay. So that is the advantage of linking with the alpha stem cell clinic, which this type of facilities are not really available in other parts of the country. So this is a unique added value of using alpha stem cell clinic for carrying out these type of treatments.
Great. Dr. Talib, thank you so much for joining us today and talking to us about these clinical trials. We enjoyed it.
My pleasure. Okay. My pleasure, Kevin.
Now we're going to bring in two other CERM science officers, two fabulous CERM science officers. Dr. Ken Fitzgerald, looking splendid there, and Dr. Kelly Shepard, who I believe is going to be joining us momentarily. Kelly, can you hear us?
Yes, I hear you.
Great, good to see you. Love the background. I love those stem cell images, they're so sharp. So we're going to be talking about we just heard from Dr. Talib talking about the clinical trials that we've been funding. We've also funded a number of projects at a much earlier stage or earlier stages of research, and we're going to be looking at those now and why those are important as well in trying to deal with something as immediate as a pandemic. And if you will, I'd like to begin by looking at a video from Dr. Gay Crooks from UCLA. Yeah. Sorry. Stuart Lipton snuck in. As he's wont to.
Our goal is to find the best way to stimulate the immune system to fight the COVID-19 virus, and blood stem cells are key to solving this problem. To develop a vaccine, we want to use a part of the immune system called dendritic cells, which help process the virus in our body into smaller pieces called peptides that can stimulate the immune system. Unfortunately, the most powerful types of dendritic cells in the body are incredibly rare in the blood, too rare to use for these experiments. So we found a way to take blood stem cells and generate millions of dendritic cells in a dish to discover the best viral peptides to stimulate the immune system. These peptides could be used in a vaccine.
Great. Well, I hope the people who are funding in their research are more skillful at that than I appear to be at Zoom, but we'll get through this. So Kelly, a number of the projects we're funding targeting vaccines, but there are also a number of vaccines that are already in clinical trials. What's the advantage of us funding this kind of work at an earlier stage?
So, you know, we have a lot of experience with vaccinations, and they can be very effective at preventing disease. But as with anything that's a new area of science, figuring out a way to make the vaccine the most effective and that can help the most people is always going to be a challenge. Every virus, every pathogen is a little bit different, and everybody's immune system is a little bit different. So kind of the first line approaches, and these are the ones that you are seeing in clinical trials now, are kind of going with an obvious strategy, which is to vaccinate people with portions of the pathogen, which is in this case pieces of the COVID virus or components of the COVID virus, and letting the body do the work. Now, what is this work? Well, an immune response, there's really kind of two pathways that it falls into. One is the humoral response, which is antibodies. And we hear a lot about that. In fact, Sohel discussed a characterization of the antibody response in convalescent plasma therapy, because not all of the antibodies that our bodies produce are the same. Some of them bind very strongly to pathogens, some of them bind weakly. Some of them neutralize the pathogen. That is, they bind to the virus and prevent it from being able to infect cells to begin with. And that's obviously the kind of response you want. But not all antibodies are equally effective as that. And once the virus is able to get into a cell, well then, you need another form of immunity to help clear those out. And that's our cellular immunity. So the other side of our immune system is the cell immunity, and that's affected by special cells, especially T cells, which when they recognize an infected cell, they actually kill it. And not only do they kill it, but they create an army of themselves so that they can do a wholesale onslaught on the virus. And this is also the cellular response is what allows your body to have a memory, so that if you recover and you're infected with this pathogen and again in the future, you have cells at the ready. And part of the cells that are at the ready include a form of a stem cell, so that you can rapidly respond and produce more of these cells that will recognize the pathogen. So the approaches that CIRM is developing are really approaches where stem cells are either used as tools, or approaches are used to try to take advantage of the stem cell component of immunity to make a more effective vaccine. One of these approaches was in the video you showed earlier with Dr. Crooks. She's actually created sort of like almost like an immune system in a dish, where she can test different pieces of the pathogen and identify the T cells that respond to that, and then she can identify what pieces of the pathogen those T cells are responding to, and then you can use those to vaccinate people in the future, and perhaps they'll be more effective. We have a couple of other approaches that are also trying to achieve a stronger form of cellular immunity. In one case, Dr. Albert Wong at Stanford, he's actually combining pieces of a viral protein with T cells from individuals of different ethnic backgrounds. And this is important because cellular immunity doesn't just depend on the interaction between a T cell and the pathogen, but there's also a piece of your own body, the proteins that are expressed on your cells, the context of which plays a role in that response. And people of different ancestry and different genetic backgrounds have different varieties of these proteins. So they sometimes respond differently. So he's actually using stem cells to derive T cells from patients of different diverse populations and test them in vitro to see which pieces of the spike protein are most effective, so that vaccines can be developed more efficiently. And finally, we have one other vaccination project being done by Dr. Song Lee at UCLA. And he's actually using a biomaterial in combination with pathogen proteins to try to stimulate the memory response. Because one thing about vaccination is, although they can be very effective, they often don't work so well in elderly people who actually lose some of this memory response as they age and as the cells become older, and also people with certain pre-existing conditions. So by developing a vaccine platform that can actually stimulate this memory T cell response, it's hoped that you'll be able to actually boost the ability of elderly or other affected people to respond effectively to a vaccine.
I want some of those memory cells. I could certainly use them. Can the vaccines or these approaches be also used then for other diseases if they're effective against COVID, if we learn enough from them?
Absolutely. So in these particular projects we're funding, our investigators are using pieces of the coronavirus, SARS-CoV-2, in order to identify the most effective responses against those particular pathogens. But you can actually do it with other forms of disease and even, cancer proteins, anything where an immune response could be helpful. And in fact, one of our projects that is developing a COVID vaccine, Dr. Albert Wong from Stanford, he's actually adapting a vaccine approach that he developed to go after glioblastoma, which is a brain tumor. So yes, absolutely. These platforms should be useful, not only for trying to identify better COVID-19 vaccines, but also it has a potential to address other types of both infectious diseases as well as possibly cancers.
Great, thank you, Kelly. We'll come back to you in a minute. We're going to switch gears now and talk to Kent, Dr. Fitzgerald. But first, I'd like to show another video, if I can. And this is Bridget Gompertz, who's another researcher at UCLA. Dr. Jinhua Hu just keeps trying to get in there. Amuse yourself, people, while I'm doing this. Thank you very much. Here we are.
Hi, my name is Bridget Gompertz, and my lab studies lung stem cell biology. A few years ago, we developed a lung stem cell derived model we call a lung organoid, which closely resembles lung tissue. And when the COVID-19 pandemic hit, we realized that our lung organoid could be used to study the infection. So we've partnered with Vaiti Arumughaswamy, a virologist at UCLA, who's infecting these tiny lung organoids. We've found that the infection of the organoids closely resembles the viral infection seen in the lung of patients. And this is allowing us to study thousands of compounds in order to try and find one that will prevent infection and also reduce lung tissue damage. Currently, we're screening thousands of FDA-approved compounds with the hope that these could be repurposed quickly to get to the clinic.
So Kent, so a number of the projects we're funding take a kind of slightly similar approach, don't they, in terms of going either after the virus or trying to repair the lung tissue?
Right. Excuse me. So one thing I wanted to point out, and this addresses something that our chairman, John Thomas, mentioned. All of the awards that we're funding in this what we call discovery and translation space are 12-month maximum duration projects. So it really addresses the urgency of the pandemic. And the goal of these projects is that at the end, the people working on them will have a therapeutic candidate that's ready to move to the next stage of development. Maybe that for a discovery program that is into translational work, and for a translational program that is having initial talks with the FDA, which obviously is required to move into clinical trials. So all these projects are moving really fast. And in many cases, projects like Dr. Gompertz were fortuitous in that she already had a model in place, which is highly appropriate for testing COVID-related drugs. So two ways really that stem cells can play a role in developing and testing alternative therapeutics other than vaccines. One is they can be used to generate the experimental model, as Dr. Gompertz described. The other is that the stem cells of the lung are unfortunately the target of SARS-CoV-2 infection. These are called alveolar type 2 cells. As I mentioned, they act as the stem cells of the lung. They serve a couple functions, one of which is to produce material called surfactant, which lets your lungs function and expand, kind of a lubricant. And the other is that they help repair damaged lung tissue. And as you can imagine, being the target of the virus, these cells are damaged and killed, and that can lead to long-term deleterious effects in the lung. So one of the reasons why stem cells become relevant here is that these AT2 cells are very difficult to grow in culture. So this enables things like what we call induced pluripotent stem cells to generate lung tissue, as some of our projects have. Dr. Gompertz's project, as she mentioned, it utilizes lung organoids. Now, an organoid is a three-dimensional tissue, almost like an organ, like a mini organ. So her tissues can be considered kind of mini lungs. And they have a lot of benefits over traditional cell culture testing systems where researchers will put a drug on cells in a dish. And oftentimes these cells don't really represent the tissue that they're being tested for. And in the case of Dr. Gompertz, she mentioned she is using a large library of thousands of previously approved FDA drugs to see if any of those might have an effect on viral replication as well as lung function that she can measure in her system. And this is important because you can't take a drug and just put it right into a person. Likewise, the value of testing drugs on more artificial systems, maybe animal-derived cells or animal systems, isn't quite as predictive as you would hope. And that's where the real value of using human organoid-type tissue comes into play. We have a couple more programs that we funded in this space that are also going after more of an antiviral type approach. So if you can knock down viral replication, you can then knock down infectivity, you can minimize damage as well. So Dr. Justin Achita at USC is testing a specific compound which acts on a kinase that's known to be relevant for viral infection. And in fact, he is testing a compound that is related to a known compound that was shown to be 25 times more potent than remdesivir, which has been in the news a lot recently as being effective. So he is testing that to show activity on an IPS, so a patient-derived pluripotent stem cell lung AT2, so the infected cell in culture. Another organoid-based approach that we're funding is being done by Dr. Evan Snyder at the Sanford Burnham in San Diego. He is also screening drugs. His project is, as Kelly alluded to, testing organoids developed from a diverse genetic background of patients, so diverse ethnicity, with the hopes of getting something that will work in a diverse population. He is also using this 3D cell system. And one nuance to his project is, as you know, your lung isn't made up, well, as you know, but maybe not everyone else knows.
Oh, I don't know. But thank you for thinking that I might.
Your lungs aren't made up of one specific cell type. As everything in your body, you're made up of a bunch of different cell types. So his organoid system utilizes both lung-specific cells as well as cells called macrophages, as well as some of the vascular cells, so the cells that perfuse the lungs with blood. So that complex interaction of cells often can impact the way a drug works and ultimately how they behave in the body. So his goal is to find a drug that suppresses the severity and contagiousness of COVID. And finally, Dr. Gompertz mentioned Dr. Vati Aragaswamy. He is a great example. So he has a later stage project. He is not aiming to discover a treatment. He already has a treatment that he's proposing will work. And he's a good example of a couple things. He is collaborating with Dr. Gompertz. And this is a great sort of example of our researchers and how they tend to work together and accelerate timelines. So he has an expertise in one area, she has an expertise in another area. They're coming together to work together to push things forward. So he, interestingly enough, back early in the year, when there were only a couple dozen cases of COVID in the US, he was previously working on Zika virus, and he's a virologist. He had the foresight to get some isolation of the virus from those first couple patients in the US. And I had a phone call with him, and he said, you know, he could tell this is going to be a problem, probably something he should shift gears and start working on. So
Prescient.
I don't think he had any idea how big a problem it would be. But so he was able to shift gears with his project and start looking at antiviral drugs that may have an effect. And so he was really ahead of the game from a lot of people. And he had come up with a drug that he believes has an effect. He's shown some early, what we call preclinical data, showing that it has an effect in in affecting viral replication. So his goal with his project is to move that ahead to an early filing with the FDA. So something that's going to get it one step closer into going into patients.
Great, thank you, Ken. We're going to look at some of the other researchers we're funding now who are targeting different parts of the anatomy. Let's see. And this is Dr. Helen Blau from Stanford, who's looking at the kind of the consequences of being on a ventilator for an extended period of time and the impact that that can have on a person.
I'm Helen Blau. I'm a professor at Stanford University in the School of Medicine. And I've received funding from CERN in order to test the potential of a drug combination we've discovered that targets stem cells to improve outcomes for COVID patients who suffer from VIDD, that's ventilator-induced diaphragm dysfunction. Millions of people in the United States have now been affected by COVID, and 160,000 or more have died to date from COVID. And one of the major causes of death is that people who require being on a mechanical ventilator experience diaphragm failure. And what happens is that when they're weaned from the diaphragm, which breathes for them and takes over for their muscles, the muscle atrophies, it becomes weak, it becomes small, and it is very hard for the diaphragm to then function. Our proposed therapy could strengthen the diaphragm by stimulating the muscle stem cells that are present in the diaphragm, encouraging them to regenerate and rejuvenate the muscle fibers and restore strength.
And then we're also going to hear from Dr. Stewart Lipton. Well, apparently not. Oh, there he is. The little devil.
Hi, I'm Stuart Lipton. I'm an MD PhD at the Scripps Research Institute in their Drug Discovery Center, which you see behind me here. And I'm really proud that CERN has awarded us a grant to work on COVID-19.
Start that again.
Hi, I'm Stuart Lipton. I'm an MD PhD at the Scripps Research Institute in their Drug Discovery Center, which you see behind me here. And I'm really proud that CERN has awarded us a grant to work on COVID-19. We've made a really important serendipitous discovery. We have developed a drug for Alzheimer's disease, which is now FDA approved, that was based on an antiviral drug. We then optimized.
Are we able to see this? Are you seeing this? No. Okay, let me try that again.
I can hear it though.
Okay, well, that's a start, I suppose. Okay, okay.
There you go.
But let me bring it back to the beginning because Stuart has some great things to say. And he looks so good.
Really important serendipity.
Okay.
Hi, I'm Stuart Lipton. I'm an MD PhD at the Scripps Research Institute in their Drug Discovery Center, which you see behind me here. And I'm really proud that CERN has awarded us a grant to work on COVID-19. We've made a really important serendipitous discovery. We have developed a drug for Alzheimer's disease, which is now FDA approved, that was based on an antiviral drug. We then optimized the drug for Alzheimer's disease. And then when COVID-19 came along, we realized, gee, we've made it better for the brain. Maybe we've made it better to fight the virus. And lo and behold, through CERN funding, we've begun to screen those new drugs against COVID-19, the virus called SARS-CoV-2, and it stops the virus in its tracks. So we've done screening in a dish. Now we're about to move into animal models to prove that it really works before we can do human trials. Thank you to CERN for this.
And thank you to Stuart for showing up. So Ken, when we talk about kind of all the different areas that we're focusing on, it seems like a lot of this research is actually was already in place. And it was just a kind of a quick response from the researchers to kind of pivot. You talked about that ability to pivot that
Right.
really made it something that could be effective against the virus.
Right. Yeah. So in addition to the obvious ways to go after COVID, such as antivirals, vaccines, etc. There are a lot of secondary effects that were alluded to by Dr. Blau. And so we have, obviously being the Regenerative Medicine Institute, a lot of our funded work really aims to regenerate tissue through endogenous mechanisms like stem cells. So many of our PIs that we fund really, they're all very creative, and they were able to say, hey, in Dr. Blau's case, she's working on muscle stem cells, and she's also a physician, so she knows that ventilators cause diaphragm damage. The diaphragm's the muscle that enables you to breathe, moves your lungs. And so if you're damaging that, and she has a therapy that she believes is going to help restore that diaphragm muscle, it's an obvious pivot, right? Takes some foresight, you have to be thinking ahead. Dr. Lipton, as he described, the same thing. He's a very accomplished drug discoverer, and he has a certain area that he's focused on. And he mentioned his compound, his FDA-approved compound for Alzheimer's disease. Well, it turns out, and he knew that the coronavirus family, of which SARS-CoV-2 is a member, has some of the same proteins on it that his drug interacts with in the brain. It also interacts with these proteins on the virus. So, maybe he should test that and see if he has an effect. It also was quite fortuitous in his case, in terms of secondary long-term effects of COVID-19 infection. There are a number of downstream long-term effects that we're starting to see emerge, right? So one of these is diaphragm damage. I was looking through, the WHO has published a list of documented secondary effects. These include things like obviously unresolved lung damage, heart damage, which we've heard a little bit about in the media. The brain and the nervous system are definitely affected. We're learning there are effects like loss of smell, cognitive impairment, which is similar to Alzheimer's disease. There's also an emergence of Parkinson's-like symptoms. I think that people may be predisposed to develop later Parkinson's after COVID infection. So if you can't stop people from becoming infected to begin with, you need to then also maintain and try to resolve some of these long-term effects. So we have funded 3 programs in that addressing that need, one of which is Dr. Lipton, the other was Helen Blau. What she didn't mention is that in addition to ventilator-related diaphragm damage, really it's a dangerous thing. About almost 30% of ventilator patients develop pneumonia, and of those, up to a half of them die from pneumonia. So you see people, you hear about people dying from COVID infection acutely, but long term there may be effects that we would like to avoid. The third project that you didn't have a video for is Dr. Karen Chrisman at UC San Diego. She is also going after ventilator-induced lung injury. However, this is more focused on the lung itself. So she has a product that she has developed, which is it's a form of extracellular matrix, which is like a framework that your cells exist in, that your cells secrete. Collagen, which most people are familiar with in your skin, is part of your extracellular matrix. It also contains a number of other proteins which serve to maintain and kind of keep cells healthy in their proximity. So her product is an isolated, purified form of extracellular matrix that she has previously shown in a cardiac project was able to inject into the heart muscle and recruit endogenous stem cells or recruit repair mechanisms to the damaged tissue. Now, she and her project for COVID is testing to see whether or not similar product, which is an extracellular matrix specific to lung tissue, might be able to recruit repair mechanisms into damaged lung tissue and avoid some of the long-term lung effects.
Great. Thank you, Kent. We're going to switch back now to Kelly. And the last video, honestly, I promise you this will be the last video. Well, in fact, we won't have that last video. There we are. We can only hope that stem cell research progresses faster than my technical abilities. But here is Dr. Jinhua.
Hello, everyone. My name is Jianhua Yu. I'm a professor at City of Hope. City of Hope is using a very unique approach to hopefully treat COVID-19 patients. We are taking stem cells from umbilical cord blood and turn them into chimeric antigen receptor natural killer cells, also known as CAR NK cells, to attack the coronavirus. The goal is to deploy these CAR NK cells to specifically target the cells infected with the coronavirus and clean the virus from patients' bodies. This treatment can be used as soon as patients become infected with coronavirus. Importantly, this treatment could be crucial in treating patients who are at an increased risk of developing serious complications from COVID-19, including cancer patients and all the elderly population.
So Kelly, talk a bit about these other approaches that Dr. Jinhua Hu was kind of exemplifying, the kind of different ways that we're funding to try and attack the virus.
So Dr. Yu was talking about a form of the cell therapy, and specifically an immunotherapy, where we're actually teaching our immune system to go after the virus. So I feel like we're coming a little bit of full circle here, because I started off by talking about how our immune system can be treated in a way to help prevent us from getting a disease to begin with. And now we're talking a little bit about how we can exploit our own immune systems to be better at fighting immune diseases. This is also tying directly into some of the work you heard about in clinical trials from Dr. Talib, who came on right before myself and Kent, who was talking about some of the types of cell therapy candidates that are being tested for treating COVID-19 that are already in clinical trials. So how can a stem cell or a stem cell derivative help us fight an infectious disease? Well, one way is it can go after the damage caused by the disease. And Kent talked a little bit about some other types of ways that some of our investigators are looking at trying to treat or regenerate diseased tissues, using small molecule drugs, for an example. But there are also certain cells that have properties that can be conducive or promoting to regeneration or repair. One of them is the MSC cell, which Dr. Matei is testing in his clinical trial, which is these cells have properties that sort of dampen inflammation. So the SARS-CoV virus, not only does it directly kill cells, but it also causes damage to associated tissue because of, sometimes there's an inflammatory response that causes changes in the cells around it. It can cause inflammation and difficulty in breathing. Now, this inflamed environment can also make it difficult for cells to heal. So one rationale for a cell therapy is that a cell with some of these anti-inflammatory properties can kind of calm the environment and create a more conducive situation for cells to heal themselves, or for a regenerative therapy to actually try to regenerate or replace those cells. You also heard in the clinical trials the use of natural killer cells, which are another type of immune cell that naturally go after viral infected cells or other cells that are abnormal in our bodies. Now, Dr. Talib mentioned that in patients that are suffering from certain disorders, our normal levels of these natural killer cells go down. So in that trial, the thought is by actually giving a boost to people with additional natural killer cells, in that case derived from cord blood stem cells, could be of benefit. So now coming back to these discovery stage projects which are in a much earlier state, we're talking about the next generation of approaches. The ones that are in trials now, we're kind of close and ready to go, or could be tweaked in a way so that they could be deployed quickly. And that's what we have. But what we want to have are things that are like smart bombs, basically. So we're trying to take what we've learned from our stem cell biology and regenerative medicine and create cell therapies that are more specific and can do the job more efficiently, taking advantage of the power of our own immune system. So we have a couple of projects like this in our discovery stage. One of them you just heard about from Dr. Yu. He's taking cord blood-derived natural killer cells, which is in some ways similar to what is going on in that clinical trial that was discussed earlier, but he is engineering them to specifically go after the coronavirus. He's genetically modifying these cells so that they will have a receptor that will cause them to target those cells and then kill it, basically. So that's one approach. We have another approach from Dr. Chaudhuri at USC that is taking a similar approach in that he's also engineering a natural killer cell with a special receptor to go after SARS-CoV. However, in that case, rather than deriving the natural killer cells from cord blood stem cells, he's deriving them from induced pluripotent stem cells. So the source of the natural killer cells might affect their activity, and we don't really know which source is going to be best. There's advantages and disadvantages probably for a lot of these situations, and we have to explore them to figure this out. One thing about induced pluripotent stem cells is you won't necessarily have to go back for more cords to make more. Induced pluripotent stem cells are a type of stem cell that is virtually immortal. You can expand them indefinitely. There are some tricks that are involved, which is also an area of active research at CERN. So this is another example of where a lot of the things that we've funded in the past are coming together to try to help bring forward a better and newer technology for the future. And finally, we have one other cellular therapy approach in our discovery stage from Dr. Lily Yang at UCLA. And this is yet another variation of an immune cell that we're trying to train to do a better job when given back to fight infection. Now, we've talked a lot about the immune system today, and I have to say it is very wonderful and complex, and there are many, many different cell types, some of which are still being discovered today, actually. It's a very finely orchestrated organ, basically, and there's still a lot we have to learn about how to use it most effectively. And so in Dr. Yang's grant, she's looking at a special type of cell called an invariant natural killer T cell. This is different than the natural killer cells that I've just talked about, and it's different than T cells, which I talked about earlier, which are important for fighting infection as part of our natural cellular immunity, but which are also being exploited in clinical trials to when they're engineered to go after cancer cells. So in this case, she's looking at this this other type of killer cell, which has some slightly different properties and recognizes antigens in a slightly different way. And she's doing some genetic engineering to try to make them more effective. And this is actually a type of project that she has been working on towards addressing a cancer. And now she's able to try to repurpose this cell in a way to make it go after an infectious disease. So you had asked me earlier in our conversation about whether or not these approaches can be applied towards different diseases. And the answer is yes, there are many cells that we can try to exploit within our own immune system to make them more effective fighters. In this case, there's some genetic engineering that's involved to try to get them to go after a specific target and to try to make them last longer and be more persistent and to work better in all patients, and whether they're elderly, whether they have different ancestry, what have you. So these are kind of some of the next generation approaches that are being developed now. But as Kent said, they have very short timelines, very aggressive. They're all in a way being repurposed from therapies that were being developed with stem cells for tackling other types of diseases. But they're basically serving as a platform that we can then try to repurpose so that we can act rapidly to address new situations we find ourselves in, whether that be with a pandemic or whether it's to fight a cancer that we haven't been able to find any effective therapies for in the past.
Great. Well, Dr. Kelly Shepard, Dr. Ken Fitzgerald, thank you so much. It's always a pleasure to hear you explain this complex science in really simple, easy to follow ways. Really appreciate you being here today. And finally, we've been saving the best for last, and he paid me good money to say that. We've seen a lot of progress in stem cell research over the last few years, and we've been talking about that just now. But some of the most important tools, some of the tools that have helped us get to this point are now facing some restrictions or potential restrictions. And so our colleague, Dr. Jeff Lomax, is here to talk about that. Welcome, Jeff. Jeff, can you hear me? Can you hear me? Yeah, you're on. Yeah. Welcome, Jeff.
Okay, can you hear me? Sorry.
Yes, I can hear you.
Okay, there we go. Apologies. Happy.
Can you hear me?
Yes, I can hear you.
Yeah, you're on.
We're good. Happy Stem Cell Awareness Day, by the way. And yeah, so a little bit of an, I guess this is the ironic twist moment in the conversation. So a lot of the things that you. Okay, can you hear me? Sorry. Okay, there we go.
Are you?
Apologies. Happy. Yes, I can hear you.
Weird.
We're good. Happy Stem Cell Awareness Day, by the way. And yeah, so a little bit of an, I guess this is the ironic twist moment.
Do you have YouTube on, Jeff? Well, we appear to have lost Dr. Lomax for a moment. I'm hoping that he'll be able to join us again. We're going to be talking here about some of the potential new restrictions on both fetal tissue and embryonic stem cell research. These are two of the tools that people have been using most commonly over many, many years. Fetal tissue research has already been, there's some restrictions been imposed on federal funding for that. And there's also some moves in Congress to impose some restrictions on federal funding for embryonic stem cell research. Can you hear me now, Jeff?
I think we're back. Sorry about that. There we go. It was, I think it was competing versions of Zoom fighting with one another. A little bit like COVID and the immune system. You know,
nice pivot.
It's a back and forth there. So I heard your introduction, and I'll just go with how I was going to respond 45 seconds ago. It was really interesting. So from my colleagues, you've heard a lot about these model human organ systems. You've talked about the lungs, the immune system. They're all, great tools for exploring, how we can develop treatments for COVID or how we develop vaccines. But what's really interesting is a lot of these tools, these organ systems, are made possible by something called human embryonic stem cells, which probably people have heard about. And over the years, there's been quite a bit of back and forth about, whether they should be used. And it was actually interesting. Recently, Senate Republicans circulated a letter encouraging the president to abolish President Obama's policy, which created a list of ethically created human embryonic stem cells for use in exactly this type of research. And incidentally, many of the human embryonic stem cell lines on this list were created in California by our CIRM awardees. So we are in this kind of peculiar moment where this sort of vital research tool is actually, being threatened by, politics in Washington, basically.
And also when we look at, say, fetal tissue, I mean, that's been used for decades and decades to help develop some of the most important vaccines that we use today, rubella, chickenpox, and develop therapies against things like hepatitis A. So really, really important. So it seems ironic at this point that we're going to be maybe closing the door on something that would be tremendously useful in helping tackle or address COVID.
Yeah, so again, similar point. Again, the fetal stem cell lines are perhaps the most durable and longest serving tool available in vaccine development. I mean, they've been around since the 70s. They're so important that they really are the standard by which all these treatments are potentially evaluated. So absolutely central. And if you had to sort of come back today and develop a sort of a new tool along the lines of kind of what Kelly was describing, using these new innovative techniques to come up with something new, you would actually have to go before a committee that was put together by the Trump administration to get permission to do that work if you wanted to use a fetal stem cell line. And again, just to kind of really bring this back to home, that committee has already rejected proposals from California-based universities that were recommended for funding by the NIH peer review process. And the peer review process, as you may be well aware, is really the leading experts looking at a scientific proposal. And it's a very difficult process. On average, less than 5%, 10% max proposals get through. This got through saying, oh, this is very important scientifically, but we're not going to allow it simply because you want to use a tool that we, for whatever reason, don't feel like you should have the opportunity to use.
And it seems, I mean, obviously one of the advantages of having CERN is that we can avoid these restrictions. So since 2004, when we were created, we were able to fund things that, and bring researchers here to do work here that they wouldn't have been able to do in other places.
Yeah, well, it's funny you bring that up as well, because ironically, one of the proposals I was alluding to really came about because of years of CERN funding and the fact that we create a safe harbor for all types of research. And, as folks may be aware, CERN has had to greatly scale back its funding until our future becomes more certain. So as a result of not being able to go back to CERN to really complete that work, they went back to the federal government, and that's where the roadblock I described that they hit. So at the moment, some very promising work that we've always allowed won't continue until there's a pathway where they can move forward. And at the moment, they're blocked by this committee.
And we've already seen that a number of the vaccines and potential therapies that have been developed against COVID have involved the use of fetal tissue or the cells from these tissues.
Yeah, it's interesting you bring that up. It's a funny story. I don't know if you noticed, or the audience may have heard the news that President Trump was treated with this remarkable new therapy and was very excited about it. Well, who would have thunk, again, the fetal stem cell line you described earlier, again, which was developed in 1970, was very instrumental in the development of that treatment. So it's kind of ironic to think that a stem cell line created in the 1970s would be treating the president, would be instrumental in a treatment given to the president of the United States in 2020. And I think it just goes to illustrate the importance of, well, the rule, you don't know what you don't know. And anytime you start creating artificial restrictions on invention, knowledge, and science, nothing good could come of it. You'll only limit what the future potential.
Great. Jeff Lomax, thank you so much for joining us today. Really appreciate your thoughts on this.
Thanks for the opportunity. Appreciate it.
And so that's it. That's our program for today. I'd like to thank all of you for joining us and for sticking around, for putting up with my technical glitches. And as I said, I hope that our researchers are better at doing their job than I am at this new technology. But we'll get there. And thank you again for joining us on Stem Cell Awareness Day, and stay safe out there.